I'm Mike Huckman, and I'm here with Sanofi Chief Scientific Officer, Dr. Gary Nabel. Dr. Nabel, nice to be with you.
My pleasure.
Trispecific antibodies. Explain to us what that is and how it was invented by the scientific team, your team here at Sanofi.
A regular antibody is Y-shaped, and the two arms of the Y recognize the foreign protein. We developed at Sanofi, before I got here, a technology that allowed us to take those arms of the antibody and do a double stack. And what that allowed us to do was to make a bispecific antibody. And what we then did is we took that molecule and we said, how could we modify it in a way where it could have even more breadth, see more things? We said, what happens if I take the double stack, the two arms, and I combine that with a single arm that's different? And I do that with a little bit of a molecular glue that brings them together. Then I could have two stacks on one side, a third stack on the other side, but it still looks essentially like an antibody. And that's the origin of the trispecific antibody.
To what end, Dr. Nabel? What does that get you?
Where we started our investigation of the technology was with HIV. There are a few conserved sites on that virus. And what we knew is there were 3 very good antibodies that did recognize these different sites. The problem is that if you were to try to develop 3 antibodies to neutralize HIV, it would be very expensive to make 3 antibodies, number one. The clinical trials would be very difficult because you'd have to mix and match the different antibodies. They all have different half-lives.
So put them all in one.
So you put them all in one, yeah, and it simplifies the things. You have one product, it can recognize all of these different targets, and then with one treatment and one product, you can now go after these multiple targets. I will say the other advantage of the technology is that, I mentioned in the antibody molecule we have the Y-shaped, and at the ends of the Y we recognize the foreign target. But there's other business that's going on in this molecule at the base, which is holding these two Y-shapes together.
So it's not just reaching out and grabbing and recruiting things.
Exactly. There are what I would call protein activators at the base that can pull in other parts of the immune system. And we can manipulate
to fight
those.
Whatever the
To
bad
fight
actors.
infections, to get rid of tumor cells. So they also recruit other types of cells.
So it sounds like it's flexible, utilitarian, multi-purpose.
Multi-purpose. It really is kind of a plug-and-play cassette type operation. We need to make sure that it's well tolerated in people.
Safe.
It's safe, and that it doesn't engender or elicit an immune response against the molecule.
What, if anything, makes the Sanofi trispecific antibody scientific platform unique or special?
Well, what I will say is that, at least at this point in time, we're leading in the effort to move this into the clinic. We'll probably be the first trispecific in the clinic. We're due to be in patients later this year. I don't know of another company that is that far along.
How challenging, how difficult is that engineering that you've described?
The spectrum of what we do is incredibly complex, and we literally had a village. We had people from the Frankfurt hub, from the French hub, from the US hub, from the Breakthrough Lab, all of them working collaboratively to do something which I don't think anybody or any individual lab could ever have done on its own.
Dr. Gary Nabel, the Chief Scientific Officer of Sanofi, pleasure to be with you.
Thank you. My pleasure.